Hello, everyone, and welcome to today's webinar, Rapid Detection of Carbapenemase Activity in Carbapenemase-Producing Organisms, CPO, using MALDI-TOF. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is hosted by LabRoots and sponsored by Bruker. To learn more about our sponsor, please visit bruker.com. Now, today's presentation is interactive, and we encourage you to participate by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Now, without further delay, I would like to now introduce today's speakers. Dr. Chunye Liu, Antibiotic Resistance Testing Laboratory Supervisor, Bacterial Diseases Section, Microbial Diseases Laboratory, California Department of Public Health. And Vida Mofidi, Senior Public Health Microbiologist, County of Orange Healthcare Agency, Department of Public Health, Santa Ana, California. For complete biographies on our speakers, please click on the Presenter tab at the left of your screen. Chunye, Vida, it's so wonderful to have you here today. You may now begin your presentation.
Hello all. Thank you so much for the introduction. Let's begin with a little bit of a history. Carbapenem-resistant organisms, CRO, have evolved through time. This slide gives a quick snapshot of a CRO emergence and its effects in our communities. In 1985, imipenem was the first clinically approved carbapenem drug used in the United States. In early 1990s, the first carbapenem-resistant case emerged worldwide. The discovery of the first carbapenemase-producing Klebsiella pneumoniae in North Carolina in 2001 was a turning point in the history of antimicrobial resistance. The KPC carbapenemase enzyme led to a worldwide spread. World Health Organization urged the need for new antibiotics, and CDC urged robust surveillance programs and discussion for all states in the United States to add carbapenem-resistant Enterobacterales, CRE, to reportable conditions. The CDC carbapenem-resistant organism surveillance program case definition has changed as carbapenemase-producing organisms, CPOs, became a more epidemiologically important group of MTRO pathogens threatening public health. In 2017, CRE became reportable in Orange County, and volunteer submission of isolates from all regional laboratories began. Under Title 17 of California Code of Regulations in 2019, CPCRE cases officially became a mandatory reportable condition throughout the state of California. To align with the CDC case definition, California Department of Public Health updated the Title 17 in 2022 and changed CPCRE to CPOs. Regardless of all efforts nationally and globally, in 2022, an artificial tear product contaminated with a novel carbapenemase-producing Pseudomonas aeruginosa was responsible for an 18-state outbreak that resulted in 81 cases and 4 deaths. Traditionally, carbapenem-resistant organisms were associated with infection in healthcare settings. Nonetheless, in 2023, the first domestically acquired carbapenemase-producing Pseudomonas aeruginosa was reported in Southern California. It raised more public health concern that the patient lacked international travel, but the patient's isolate carried a genetically invasive NTM gene with potential community spread. This study demonstrates that delay in the detection of a CPO can lead to the emergence of even more resistant strains and further spread of a CPO in community and healthcare settings. The objectives of this webinar are to provide background information on carbapenem-resistant organisms and carbapenemase-producing organisms, review currently available phenotypic tests to detect CPOs, introduce a STAR-Carba assay, summarize the results of a validation study, and present a CPO workflow scheme that incorporates the STAR-Carba assay for primary phenotypic testing. Let's start with a bit of background information. Carbapenems are complex subclass of beta-lactam antibiotics and are often used as a drug of last resort for multi-drug resistant infection. When the beta-lactam ring of a carbapenem binds to bacterial penicillin binding proteins, it inhibits the cell wall production and leads to bacterial death. The combination of a beta-lactam ring fused to a 5-member double-bonded pyroline ring with variable side chains in carbapenems provide broad-spectrum capabilities, offering strength, potency, and stability against a wide range of gram-positive, gram-negative, and anaerobic bacteria and against beta-lactamase enzymes. A spontaneous mutation in bacterial genome is a natural phenomenon, but critical in developing antimicrobial resistance. Carbapenem-resistant organisms are bacteria that develop resistance to carbapenem antibiotics via intrinsic or acquired mechanisms. Intrinsic resistance is a mechanism that the organism is born with the ability to resist specific antibiotics. Acquired resistance is a resistant mechanism that emerges within the same bacterial population that was originally susceptible and allows bacteria to survive in the presence of that specific antibiotics. Pseudomonas aeruginosa, CRTA, Acinetobacter baumannii, CRAB, and Enterobacter aureus, CRE, are common carbapenem-resistant gram-negative bacteria. There are several mechanisms that these organisms use to resist or reduce carbapenem effect. The production of carbapenemase enzyme separates these organisms as non-CPOs or CPOs. Gram-negative bacteria use several resistant mechanisms against carbapenem drugs. One mechanism is decreased impermeability of a bacterial cell wall due to loss or alteration of a porin channels. This mechanism limits antibiotic ability to enter the cell. Another mechanism is to increase activity of efflux pumps, which are proteins that actively expel antibiotics from bacterial cells. Another mechanism is the production of carbapenemase enzyme that hydrolyzes beta-lactam ring. Currently, CPO detection assays are designed to detect the presence of carbapenemase enzymes. However, presence of other resistant mechanisms can affect the test results. For example, overproduction of extended spectrum beta-lactamases, ESBL, and AMP-C beta-lactamases in the presence of porin loss or efflux activity. This combination could result in the carbapenemase-like activity. This is the common mechanism among Enterobacter aureus, such as Enterobacter coliaceae, and is often seen in MSM as false positives. Carbapenemase-producing organisms are subsets of CRO that produce carbapenemase enzymes. Chromosomal-mediated carbapenemase production is passed to progeny of the bacteria during cell division. This limits the spread of the gene. For example, chromosomal SME gene is specific to Serratia marcescens. Or carbapenemase production can be plasmid-mediated. A plasmid is a small piece of DNA that exists independently from bacterial chromosome. Plasmids can be transferred horizontally and spread the resistant gene across different species and even different genera. It can rapidly disseminate resistant mechanisms among patients via environment, medical devices, and hands and clothing of healthcare workers. CPOs are public health concerns because they are often resistant to multiple classes of antibiotics, leaving us with limited treatment options. They are associated with high morbidity and mortality, mostly in healthcare and long-term care settings. Although there are many different carbapenemase classification systems, we use Ambler classification system, which is a widely used beta-lactam system. The main point in this slide is that this classification is based on genomic sequence similarities and not substrate structures. As a result, in Ambler classification, beta-lactamases that hydrolyze carbapenems are grouped with beta-lactamases that cannot hydrolyze carbapenems. Class A, C, and D are non-metallo-beta-lactamases that use a serine residue as an enzyme active site to attack and hydrolyze the beta-lactam ring. Class C is the only class that does not contain carbapenemases. Class A carbapenemases are Klebsiella pneumoniae carbapenemases, KPC, Guyana extended spectrum beta-lactamases, GES, Serratia marcescens enzyme, SME, to name a few. Class D carbapenemases are oxygenase type enzymes. Class B carbapenemases are metallo-beta-lactamases that require zinc ion for their hydrolysis activities. New Delhi metallo-beta-lactamase, NDM, Verona integron encoded metallo-beta-lactamases, VIM, and imipenemase metallo-beta-lactamases, IMP, are class B types. Each class contains many variants and are continuously evolving with the emergence of new strains. Transitioning to CPO detection. The available phenotypic lab tests have limitations and are exclusively not recommended for CRAB due to their poor sensitivity and specificity. In Orange County, we performed the modified carbapenem inactivation method, MSEM, for several years. MSEM is the most common and widely used method for detection of carbapenemase production in Enterobacterales and Pseudomonas aeruginosa, and is currently being endorsed by CLSI. However, there are a number of limitations with MSEM. It is time-consuming and requires an overnight incubation period. It is unable to distinguish between carbapenemase activity and synergic beta-lactamase combo effect, resulting in false positives. Additionally, MSEM delivers poor sensitivity and specificity for CRAB isolates, proving to be an unreliable test. Another commonly used phenotypic test is CARBA-NP. This test is also being endorsed by CLSI. However, it is unreliable for testing carbapenemase production in CRAB isolates and has limitations in detecting Class D and Class B CPOs. Also listed in this slide or other FDA-approved phenotypic tests. Now let's introduce a new assay called MBT STAR-Carba and discuss how California Department of Public Health validated this assay to detect CPOs. I will pass it to Chunye for validation study.
Thank you, Vida. For the next part of this talk, I'm going to go over our validation study of the STAR-Carba assay at the California Department of Public Health. Before I dive into the validation, I will go over the principle and the general workflow of this assay first. The MBT STAR-Carba assay detects carbapenemase activity in bacteria by measuring the hydrolysis of impenem. As you can see here, bacteria that produce carbapenemase enzyme hydrolyze impenem, modifying the antibiotic structure and causing a mass shift that can easily be detected by MALDI-TOF mass spectrometry. Here is the workflow. We start with pure bacteria isolates. 3 to 5 colonies from overnight cultures were harvested and mixed with the reconstituted MBT STAR-Carba antibiotic reagents containing impenem. After incubation and centrifugation, cell-free supernatant is spotted onto a MALDI target plate and overlaid with matrix. Spectra are then acquired using the MALDI-TOF mass spectrometry and analyzed by the MBT STAR-BR module. After the data acquisition and analysis of the MBT STAR-BR module, the software displays a result overview as shown here, with orange dots represent impenem hydrolyzed, meaning carbapenemase activity detected, and blue dots represent impenem non-hydrolyzed, meaning no carbapenemase activity detected. And the results will also be shown in a color-coded report table. H means hydrolyzed and NH means non-hydrolyzed. Now let's move to the validation study. A total of 166 well-characterized Enterobacterales, Pseudomonas aeruginosa, and Acinetobacter baumannii were used in this study. 165 of them were from CDC, FDA AR Bank, and one OX48 Acinetobacter baumannii was selected from our MDL diagnostic submissions. As you can see from the summary table of isolates, we had a total of 80 Enterobacterales isolates, including 40 carbapenemase-producing and 40 non-carbapenemase-producing. The 40 carbapenemase-producing Enterobacterales include isolates capable of producing one or more of the following carbapenemase: KPC, NDM, VIM, IMP, OXA, SME, and IMI. The 40 non-carbapenemase-producing isolates include 22 that do not carry any beta-lactam-resistant genes, and 18 that carry other beta-lactam-resistant genes, but do not produce carbapenemases. We had a total of 30 Pseudomonas aeruginosa isolates, including 15 carbapenemase-producing and 15 non-carbapenemase-producing isolates. The 15 carbapenemase-producing Pseudomonas aeruginosa include isolates capable of producing one or more of the following carbapenemase: KPC, NDM, VIM, IMP, and M.P.M. We also had a total of 56 Acinetobacter baumannii isolates, including 7 that are susceptible to impenem but possess a single OXA genes. A representative collection of Acinetobacter baumannii isolates possessing one or more of the following carbapenemase genes were included: NDM, OX23-like, OX2440-like, OX48-like, OX51-like, and OX58-like. Members from OX143 family are not available for this validation. So together among the 66 isolates, 102 are carbapenemase-producing organisms and 57 are non-carbapenemase-producing organisms. All isolates used in this validation study were well characterized by breath microdilution reference method and whole genome sequencing at CDC or MDL. The carbapenemase production of Enterobacteriaceae and Pseudomonas aeruginosa isolates were validated by MSEM in MDL and CDC. Carbapenemase production based on the CDC MSEM results for Enterobacteriaceae and Pseudomonas aeruginosa were used as reference data. The carbapenemase genes from CDC or MDL whole genome sequencing results for Acinetobacter baumannii were used as reference data. I put 3 asterisks here as I want to draw your attention here. The reason we use the carbapenemase genes detected by WGS as reference data for Acinetobacter baumannii is that there is no reliable phenotypic method available for detection of carbapenemase activity in Acinetobacter baumannii, therefore no reliable phenotypic data available. I also want to bring to your attention that it is hard to find Acinetobacter baumannii isolates without oxygenes. Nearly all Acinetobacter baumannii possesses intrinsic chromosomal OX51-like family members. To make things even more complicated, not all oxygenes confer resistance to carbapenemase. Now, let's discuss the results obtained from this validation. Here is a summary of the validation results. The assay detected all carbapenemase-producing Enterobacteriaceae and Pseudomonas aeruginosa with 100% sensitivity. 47 out of 54 Acinetobacter baumannii isolates that possess one or more carbapenemase genes were detected positive by STAR-Carba. The remaining 7 isolates each possess one single oxygen were tested negative by STAR-Carba. I will discuss these 7 isolates in detail a bit later. Of the 57 non-CPO isolates tested, 51 tested negative for carbapenemase production as expected, and 2 isolates, including one Enterobacteriaceae and one Pseudomonas aeruginosa, produced discordant results. I will also discuss these 2 isolates in details a bit later. Okay, so the 7 Acinetobacter baumannii isolates included in this study each carry a single oxygen. Based on the WGS data, 5 of them possess a single OX51-like family members, OX66, 69, or 91. 2 possess OX270-like family members, OX500, and 6 of the 7 isolates are not resistant to carbapenems. Only 1 isolate, number 312, has borderline MIC value to resistant meropenem, therefore considered carbapenem-resistant Acinetobacter baumannii (CRAB). All 7 isolates are susceptible to impenem by phenotypic AST. Based on AST results, these 7 isolates were expected to be negative for detectable carbapenemase production by the STAR-Carba assay, which is consistent with our results. Therefore, these negative results for these 7 isolates were true negatives. Although we categorized them as false negative for the accuracy calculation, as WGS data was used as reference data. In real testing situations, 6 of these isolates will not meet our submission criteria for CREB testing and will not be tested. And out of the 110 Enterobacteriales and Pseudomonas aeruginosa tested by STAR-Carba, 2 isolates had discordant results. Both isolates tested negative for carbapenemase production by MSIM and no carbapenemase genes by WGS, however tested positive for carbapenemase activity by STAR-Carba. To resolve the discrepancy, the STAR-Carba assay, MSIM, and AST were all repeated. In one isolate, number 71 was confirmed to be positive by STAR-Carba, while inconsistent results were obtained for the other isolate. I just want to point it out, both isolates are non-susceptible to impenem. In summary, for sensitivity, the assay detected all carbapenemase-producing Enterobacteriales and Pseudomonas aeruginosa and the majority of Acinetobacter baumannii with an overall sensitivity of 93.6%. And 7 impenem susceptible Acinetobacter baumannii isolates included in this study each carry a single oxygen. However, no carbapenemase activity was detected by the MBT STAR-Carba assay, which is consistent with the AST data. The overall specificity of the assay is 96.5% of the 57 non-CPO isolates, including 40 Enterobacteriales, 15 Pseudomonas aeruginosa, and 2 Acinetobacter baumannii. 55 isolates tested negative for carbapenemase production as expected, and 2 isolates, including 1 Enterobacteriales and 1 Pseudomonas aeruginosa, produced discordant results. The inter and intra-assay reproducibility is 100%, and the overall accuracy is 94.6%. And now here is the summary of acceptance criteria and achieved values in the validation study, where we calculated accuracy for each category of organisms. For Enterobacteriales and Pseudomonas aeruginosa, the STAR-Carba assay had a sensitivity, specificity, accuracy, and precision of greater than 90%, therefore passed the validation. For Acinetobacter baumannii, STAR-Carba assay had performance characteristics meeting the acceptance criteria, greater than 90%, except for accuracy. As discussed before, the 7 isolates that was characterized as false negative in our study are true negatives based on the AST profile of these isolates. Therefore, we plan to implement this assay for CREB testing as needed by our workflow. So, what can we take away from this validation study? As shown by our validation data, false positive can occur due to reference assay limitations, differences in sensitivity between assays, or the possible presence of alternative or additional resistance mechanisms. And some oxygens may confer weak carbapenemase activity that may not be detected using the STAR-Carba assay. And some oxygens may not confer resistance to carbapenems and will not be detected by the STAR-Carba assay. So the conclusion of this validation study is the STAR-Carba assay is a highly accurate and rapid method for the detection of carbapenemase activity, not only for CRE and CRPA, but also for CRAB. Now I'm going to pass back to Vida. Thank you.
Thank you, Chunye. Okay, let's go back to Orange County. In Orange County, we also validated the STAR-Carba, and we did it in 2 phases. Phase 1 focused on 66 CRE and CRPA with the equal number of expected negative and positive results. Phase 2 focused on CRAB isolates, and we tested 60 total isolates, 11 expected negatives and 49 expected positives. The current CDC case definition of CPO is based on the ability of organisms to produce enzymes that break down carbapenem antibiotics. These cases are confirmed cases of enzyme production via either phenotypic or genotypic methods with or without organism identification. In Orange County, we follow the current CDC case definition for CPOs as our criteria for isolate submission. The submission criteria are listed in this slide. Via close collaboration between our Sentinel labs and Public Health Lab, volunteer submission of CPO isolates has been a great success. However, most of our clients don't have capability to perform confirmatory testing. Therefore, we accept isolates that are carbapenem resistant, except for CRPA that also has additional requirements of resistance to cefepime, ceftazidime, and ceftazoline-tazobactam combo. In 2023, we implemented the STAR-Carba assay in our CPO workflow as the primary phenotypic test to detect carbapenemase-producing CRE and CRPA organisms. This slide demonstrates how we implemented the STAR-Carba. Carbapenem-resistant Enterobacter and Pseudomonas aeruginosa with no imipenem hydrolysis activity by STAR-Carba are reported as non-CPO. An isolate with hydraulic activity is considered CPO and will be tested by hardy NG-Test CARBA-5 for further detecting carbapenemase types. Rarely, NG-Test CARBA-5 does not detect any of the enzymes since it does not cover all types and strains of carbapenemases. When this happens, we reflex to GeneXpert Carba-R to ensure that we are detecting all potential carbapenemases. By request of our state epidemiology, all non-KPC and unusual cases are sent to whole genome sequencing for further analysis. The STAR-Carba is the first assay that successfully detects carbapenemase production in CRAB isolates. Since there are no other reliable assay available to detect the specific carbapenemase types in CRABs, once CRAB isolate demonstrates hydrolysis activity, whole genome sequencing can identify the exact carbapenemase genes. Orange County implemented this assay into our routine workflow this fall. To end our presentation, we would like to summarize by presenting you with the valuable insight into a STAR-Carba assay. Let's begin with its strength. The STAR-Carba is a rapid detection method. We have been reporting our results the same day for non-CPOs and for most of our CPOs. It performs on the same MALDI microorganism identification system with simple software switch. Although this assay and in general MALDI-TOF is considered a complex level testing, the ready-to-use kit make the make this assay fairly easy to perform with minimal training required on the instrument operation. And the spectral acquisition and result interpretations are automated. This assay is designed to take multiple readings per sample, which enhances the accuracy, reproducibility, and overall reliability of the test. As we know, detecting oxide-type carbapenemase has been challenging largely due to their low-level carbapenemase activity and their diverse genetic variants. Our results demonstrate that STAR-Carba has excellent sensitivity to detect oxide-types and other carbapenemases. In my humble opinion, WooCare offers strong technical support services. During my validation study, WooCare offered me their technical expertise and answered all of my abundant questions. Now let's take a look at the limitations. This assay is intended to detect carbapenemase activity. For negatives, you're basically done. You call it non-CPO and can report. However, when result is positive, another assay such as GeneXpert, NG-Test CARBA-5, or whole genome sequencing are needed to identify carbapenemase types. The workflow we discussed in previous slides could be used as a reference. In the most cases, the result output are either hydrolysis H or no hydrolysis NH. However, once in a while, you have unclear hydrolysis results. Your lab should have a plan in place to address and interpret those unclear results. In Orange County, we repeat those, and if the result is still unclear, then we reflect to whole genome sequencing. The unclear hydrolysis could be resulted from natural deterioration of antibiotic reagents. Although the assay has multiple parameters to compensate for natural deterioration of antibiotic reagents, we see occasionally aberrant results, which needs consultation, and a repeat of those isolates may be recommended. This test is not FDA approved. Our epidemiologists in Orange County use this to categorize MTRO into three-tier system of transmission-based precautions. It is at the discretion of your organization how to use this as a tool to control and prevent MTRO infection. This workflow is relatively new to U.S. market. Since the kits are coming from Germany and are held in customs for a period of time, we suggest that you keep a good inventory and order early. These kits are not cheap. However, they are more cost-effective in comparison to whole genome sequencing. Antibiotic reagents are packed as 3 in 1 pouch. If you open it, you must use all 3 in 4 hours. This means you need to have a plan to avoid wasting your product. In Orange County, we have been batching our samples, but once in a while, we will toss 1 or 2 tubes. At this point, we would like to share with you the things we have learned and the challenges we encountered during our validation process so that it can help all of you when you are validating this assay in the future. First of all, know your test plan. Think of a plan in your validation proposal before starting the study to anticipate discrepancies and unclear results. Know your samples. Understand Ambler classification and its application in this assay. As mentioned earlier, This assay will detect beta-lactamases that hydrolyze carbapenem. If you're using frozen stock culture for your validation study, consider the selective pressure method by adding a meropenem disc to your inoculated plate and use the growth around the meropenem disc to prepare working subcultures. Know your assay. The test parameters are essential to follow: timing, rotation speed, humidity, inoculum size, and drying time is crucial to produce an optimal result. STAR-Carba and ID can both be done on the same target if using MALDI direct method. If using the extended direct method, formic acid will denature antibiotic solution. Therefore, the ID must be performed on a separate target. And lastly, know your instrument. Prior to your validation, calibrate your instrument with ACS antibiotic calibration standard. It is different than BTS that is used in routine MALDI-TOF testing. In conclusion, we would like to acknowledge Bruker team for providing technical support, to name a few: Gangi, Desiree, and the development team in Germany. Also, we would like to appreciate the support of the leadership at Orange County Public Health, LA County Public Health, and California Department of Public Health, as indicated in this slide. This project was supported by epidemiology and laboratory capacity for prevention and control of emerging infectious diseases, antimicrobial resistant laboratory network, and SHARP fundings. We appreciate their support. And we also would like to thank Bruker for sponsoring this webinar and thank all of our audiences for taking the time to listen to this presentation. At this point, we would like to answer your questions to the best of our knowledge. Thank you.
Thank you, Vida and Chunye, for your informational presentations. We do have some interesting questions from our participants that have already come in, but to our audience, if you haven't already done so, we invite you to submit your questions now. Simply type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So Chunye, Vida, we have so many questions coming in. I'm just going to dive right in. Vida, let's start with you. Have you calculated hands-on time when performing the STAR-Carba assay?
They crop isolate, considering it is like 1 hour or 60 minutes of incubation, and we'll have like 2 QC organisms. Usually it takes about 2.5 hours to 3 hours from the start to reporting. And when we have the CRE and CRPA, depending on the average of 5 to 10 organisms, it is shorter incubation time. But for non-CPOs, it's about 2 to 2.5 hours from the beginning to end to report. But for the CPOs, because it also requires another testing like the NG-Test CARBA-5, we usually, it's about 2.5 hours to 3.5 hours to start the test and finalize our report. So considering that there is an additional reflex to a different testing. So it's pretty much from the beginning to end. It takes about 3 to 4 hours, and in one day we're done.
Thank you, Vida. Chunye, let's come over to you for this next question. The 7 Acinetobacter baumannii mentioned, I believe in slide 17, are they CPOs per CDC guidance?
So based on my understanding, I do not think CDC has a clear identification of what is a CPO. They do have definitions what are considered carbapenem-resistant Enterobacteriales, carbapenemase-resistant Pseudomonas aeruginosa, or carbapenemase-resistant Acinetobacter baumannii. So basically, according to their guideline, we need to report all these organisms, Enterobacteriales, Pseudomonas aeruginosa, Acinetobacter baumannii, that produce carbapenemase by phenotypic method, either MSEM or Carba-NP, or carbapenemase gene detected by molecular methods. So if we use that, those isolates do have carbapenemase gene, which is the oxygen, detected by a molecular method, which is the WGS. Yeah, that's the best I can say. Yeah.
Excellent. Well, thank you, Chunye. And I'd like to ask this next question to you as well. How many organisms are needed for the STAR-Carba assay? Is there a recommended McFarland number? And is it important to use the exact amount?
I don't think you need to measure McFarland standard to ensure the precise bacteria concentration used. In our protocol, it's as long as you adequately fill one microliter inoculation loop with 3 to 5 colonies, depending on the colony size, but you need to make sure you are adequately filling a 1 microliter inoculation loop. That's all you need.
Excellent. Thank you so much. Vida, let's come over to you for this next question. Can the STAR-Carba kit quantify the level of carbapenem resistance, or is it solely for detection purposes?
This assay is a quantitative, is a qualitative assay. So it basically just does the detection. It is for the detection purposes. It does not say how much of that organism is resistant. So for that, you really need to have like a MIC or disk assay to really quantify it. So that's the answer to that question.
Very good. Thank you so much. And Chunye, let's come to you for this next question. Let's see. Let's start. Could it cause infections and diseases with this strain with just one OXA?
So this is my general understanding. Antimicrobial resistance genes usually does not change the pathogenicity of a bacteria, which means it does not make the bacteria more or less likely to cause disease, although it may, of course, it may affect the bacteria's ability to survive in the host.
Very good. Thank you. We have some more questions coming in here. Chunye, let's go with this one as well. Why were the isolates sensitive even when the oxygenes were present in the isolates? And is the enzyme not producing in them, or the encoded enzyme is inactive in them, or could there be any other reason?
That's a really good question. You kind of answered your question here already. Yes, it could be because the carbapenemase is just simply not expressed from those oxygens because of some mutation, or it's because of the surrounding genome context or some other reasons, or it could be it's expressed but it's not a functional carbapenemase due to mutation. Or some other reasons. Another possibility could be the carbapenemase expressed is just really weak, so it's just not enough to hydrolyze the impenem.
Very good. Thank you. And I have some questions coming in. So Vida, Chunye, just let me know who would like to answer these questions. Our next question, what are the key considerations to be made when utilizing MBT STAR-Carba assay?
I can
I
start.
can answer that.
Oh, go ahead, Vida.
Go ahead. Okay, I can answer this question. The key consideration, if you want to have my experience and my understanding of this assay, the most difficulties in the beginning I had was really understanding the Ambler classification. And once I understood what this assay really detects, then it was much easier for me to analyze and understand what really I need to report. So the suggestion I have for my audience is to really know those 4 things that I said in my presentation, the lessons learned, and specifically number 2, know your samples. Also, the ASC, the standard antibiotic standard calibrations, I really didn't know that the instrument needs the calibration with that. So at the beginning, I had a lot of unclear results. And once I understood that it needs to be calibrated and aligned, the laser has to be aligned with that assay, then my results were really clear. I had a very good positives and I had good negatives and very few unclear. So that is my suggestions to my audiences.
Chunye, would you like to comment on this as well?
Yeah, I can add a bit on top of what Vida mentioned. It's just when some of the key consideration if you should use this assay, implement this assay in your lab. So of course you have to think of your workflow, if how this assay can be built into your workflow. Another big consideration is the cost, and we do need the MALDI-TOF mass spectrometry. So this machine is very expensive, although the reagent also right now the price is down some, but still I think it's still quite expensive. So the cost is a consideration.
Thank you, Chunye. I'll have you answer this next question as well. Is it possible to classify carbapenemase into A, B, or D, or only carbapenemase activity in general?
So this phenotypic assay, unfortunately, can only tell you if carbapenemase activity is detected or not. It cannot tell you a specific mechanism. Is this class A or B or D carbapenemase? In order to do that, you will need to do molecular test.
Excellent. Thank you. And I'm going to wrap with this final question, and I'm going to make sure we don't need any clarification from our submitter. Do any carbapenemase lead to the same product formation from hydrolysis? So would they be confident to detect through MALDI assay? Do we need any clarification on that question?
Yeah, I'm not sure I understand this question. Some clarification would be nice.
Okay, so our submitter, if you could resubmit your question, and we will get that question answered via the email address that you provide. I'll end up wrapping with this final question. How many spots do you test per sample on the MALDI target?
Can I answer that question?
Sure can.
Okay, so for each sample, you need 2 spots. So after you have your inoculum, 2 spots for your negatives is the first, and then 2 spots for your positives, and then after that, all your samples get 2 spots next to each other. for the ACS, which is your standard, which always read first, you only need 1 spot. And the thing about the STAR-Carba, it actually tells you where on a target you need to have your spots planted, so or targeted. So it's really nice. It just the map is given to you.
Excellent. Thank you so much. And we will wrap again with this final question. I'm trying to squeeze in as many as we have time for here. So for CREs, what do you think of performing the CARBA-R before the STAR-Carba? And Chunye, I'll let you answer this final question.
Thank you. So for CREs, so first the CARBA-R, they detect the big 5 carbapenemase genes. I think there are some considerations. You can definitely do CARBA-R first, and if it's CARBA-R positive, and then you don't need to do any, you can just report out. You don't need to do any further test. But if it's STAR-Carba, I'm sorry, if it's CARBA-R negative, then you can do STAR-Carba and to see if it's because of other carbapenemase beyond the big 5, you know, in the organisms.
And I can also add a little bit of information on this one, because we thought of it that way, that we go ahead and do all the detection of the types first. And we also had a lot of negatives. So we decided that to do the detection first, and whatever that was non-CPO would have been reported right away, and then we would wait for the typing. So it really depends on how you implement the in your workflow, and what is your infectious disease infectious disease institution is really needing this assay for the detection of the, helping the community. So it really depends on overall your decision as a team with your infectious disease, with your laboratories, with your institution. So you need to find that.
Thank you, Vida. And thank you, Chunye, both of you, for your time today and for your important research and sharing your expertise with us. Before we go, I do want to make sure that our audience knows that today's webcast can be viewed on demand, and Labrets will alert you via email when it's available for replay. That on-demand viewing is unlimited, so we encourage you to share that email with your colleagues and your friends who may have missed today's live event. I also want to thank our audience for joining us today and for their interesting questions. We had quite a few questions we were unable to answer. And additionally, if you have more questions during the on-demand period, submit them. They will be answered via the contact information you provided at the time of registration. Now, I do want to bring our audience's attention to a survey that will pop up in the middle of your screen when we close. We encourage you to participate, and it does help Bruker improve their webinar program and get feedback from you. So thank you again for joining us, everyone. Vida, Chunye, thank you again for your time. And as always, it's a pleasure to work with our Bruker team. Take care, everyone. Have a great day. Bye-bye.
Thank you.
Thank you.